this is the first video on the quantitative genetic course, and the first thing that I want to do is give you a really short introduction of conventional breeding, marker-assisted selection, and genomic selection. So I think that most of you are familiar with conventional plant breeding, where we use some parents to generate new populations, and in early generations we started to do phenotypic screening to select the lines that look better, and we advance those lines to further generations, and then we got data in the field or in the greenhouse with reps, and basically we do that phenotypic selection. On the other hand, with marker-assisted selection, you can see that the pipeline is similar, but the difference is that here in early generations we use molecular markers. So there are a lot of work to discover those molecular markers that I don't gonna mention here in this slide. But just imagine that you have a molecular marker that is good enough to select the germplasm, and like a common example is has molecular markers to select for diseases because it's kind of easy. And then we advance just the lines that have the molecular markers, and the remaining pipeline is pretty similar that the conventional breeding. So the main difference is that here in early generation, I have a really good marker or a couple of markers that are good enough to select the lines with GEM. And the molecular markers work pretty well to select germplasm in early generations when the phenotype that we're interested on is controlled for one locus or for few loci. So a common example in common bean is the anthracnose resistance. Anthracnose is controlled for one major QTL, so that make for us easy to use just one molecular marker and select just the lines that are resistant or susceptible, and remove the lines that are susceptible. But as you can see here in this graphic, every time that the trait is controlled for more loci, we are going to have more categories in that particular phenotype. And in some point where the trait is controlled for many loci, we are going to have a kind of a normal distribution. So we're not going to have a clear division between categories because we can have hundreds of regions in the genome that control that phenotype. And a common example of that kind of traits is the yield in crops, in different crops. And in that kind of traits, it's difficult to use marker-assisted selection because it's really difficult to detect all the regions in the genome that control that phenotype, and also it's unrealistic thing that we can use hundreds of markers to do marker-assisted selection. So that is why now we are interested in genomic selection. So in genomic selection, we basically have to use kind of the same data that we are using right now. So we have to plan a population and we have to collect the phenotype of that population. And additionally, we have to genotype that population. And the population that have the genotype and the phenotype, it's called the training population. And as in the training population, we have the genotype and the phenotype, we are able to estimate the marker effect of every single molecular marker that we have in this training population. And with that information, we can do two things. The first one is select the founders of next of future populations, and the good thing to use that information, that genetic value that we can estimate with the marker effects, is that we can do crosses between lines that have a really good genetic value for the trait that we're interested on, but also we can keep the genetic diversity between those lines. So that means that we can cross two lines that the generic value is good, but they are not closely, they are not, doesn't have a really close generic relationship, because that it could be a problem in a short term if you cross just the best of the best, but you just don't keep record of the lines that are or has a strong generic relationship. So that it's gonna be a problem for the future, and we're gonna discuss deeper that part in the genomic selection presentation that is in the third week. But what I want to show you here is that with the genetic value that you can estimate in the training population, you just can select the parents, and also you can predict the genetic values in some individuals where you only have the genotype information but you don't have the phenotype. So here you can do selections just with the genotypic information and select with the genetic value. And here you can see that the pipeline start again. So you do that as like a normal breeding pipeline in Common Bean. And something that I think that is really important to mention is that genomic selection is not different than conventional breeding. So here we have the germplasm, and every single breeding program needs the germplasm, and we need to generate some populations, and we need to generate the phenotype of those populations. But nowadays we have a lot of information that we can add to the conventional breeding. So we have high throughput phenotyping, high throughput genotyping, and we have also available information of the environments. So the idea with the modern breeding is that we can put together all this information, and we can do analysis as QTL mapping or GWAS and other methodologies to discover the regions in the genome that have association with the target trait that we are interested on. And also we can use methodologies as genomic selection, but all of gen basically, or the focus of all of gen, or the goal of all of gen, is improve the germplasm and start again the process of the breeding process. So as you can see here, genomic selection is just another tool that have been shown in other crops that has potential to increase the genetic gain per unit of time. And so that is why we're interested to start to use genomic selection in Common Bean. And today I want to discuss about the molecular basis of genetics, basically because as you can see, genomic selection has it's like a combination of the traditional plant breeding methodology, but also we're gonna use a lot of information in the genome and sequencing data. So that is why I just want to be sure that everybody is in the same page with these concepts. So first, we're going to talk about the genome, the chromosomes, and the genes. So the genome is basically all the genetic material of an organism, and the genome consists of the DNA. So the DNA is the molecule that contains all the genetic instructions in the living organisms, and basically control the function of the development and the growth and reproduction. And the DNA is made of four nucleotides that are adenine, thymine, guanine, and cytosine, as we can see in this graphic. And it is important to remember that adenine always pairs with thymine, and cytosine always pairs with guanine. So I just think that is important here that you remember the nucleotides, because we're going to talk about molecular markers that basically are the variations of those nucleotides across the genome. And as most of you are working with Phaseolus vulgaris, that is Common Bean, Phaseolus vulgaris has a genome that has around 540 million bases, so that sounds pretty huge, but actually it's not. Uh, if we compare the Phaseolus vulgaris genome with other species genome, we can see that a Phaseolus vulgaris genome is like between Setaria and sorghum, and Homo sapiens, the genome of Homo sapiens is like 6 times bigger than common bean genomes, and wheat has a really huge genome, so it's several times bigger than the common beans genome. And common bean is a diploid organism and has 11 chromosomes, so it's kind of easy to work with common bean because the genome is small and it's a diploid organism. So yeah, and all the information of the 540 megabases of the genome of common bean is compacted in 11 chromosomes. So basically the chromosomes contain all this genetic material that is the DNA, and the DNA is wrapped around these proteins that are the histones, and that is important because basically they maintain the integrity of the DNA into the chromosomes. And as you can see here, we have the chromosome has the centromere, and around the centromere, the region that is around the centromere, it's called the pericentromeric region. And in the pericentromeric region, the DNA is more compacted, so there are more concentration of histones, and that is important because in that region, as the DNA is more compacted, the number of genes it's lower if we compare that region with the arms that are almost in the extremes of the chromosomes. So common bean has 11 chromosomes, and here this is a paper of 2014 that was the reference genome paper in common bean. And as you can see here, this red black line is the centromere, and the gray color represents the pericentromeric region. And here there is like interesting information to share. So here the red color represents the gene density, and as you can see here, the gene density is lower around the pericentromeric region, or in the pericentromeric region, and that it's opposite to the repeat density. So the repeat density is basically the repetitive or redundant information sequences in the genome. And as you can see here, in the pericentromeric region, there are more repetitive sequences. And the blue color here represent the recombination rate. So the recombination is higher in the arms and is lower in the pericentromeric region, which makes sense because remember that in the pericentromeric region, the DNA is more compacted, it's more condensed. So that region here it's like less active than the regions in the arms. So that is why in the arms there are more genes and there are more recombinations, and that recombinations are very important, and we're gonna discuss that later. uh, yeah, so now that we mentioned the genes is the unit of inheritance and basically contain the information that we can see in the phenotype or in the traits of interest. So that is why we're interested in the genes, and that is the most common representation of the genes. So the genes have a promoter region, and basically that promoter turn on or off the gene, the expression of the gene. And that means that, for example, if the plant has a gene that confer a resistance to a particular disease, uh, that kind of genes are not always active if the pathogen is not with the plant, but with the plant, the pathogen it's in the plant, the promoter can turn on the gene, and the gene is going to start the production of proteins or the expression to cause the resistance of the plant. And then we have the exons, and the exon basically is the sequence that it's going to be expressed in the protein. And then we have the introns, and the introns are the regions of the genome or the genes that are not expressed into the protein. So here, this is the DNA that have all the region, the promoter, exon, and introns. And after the transcriptions, that is the process that the DNA become an RNA, we can see that we can mention here that this RNA is an immature RNA. And basically here you can see that we don't have the promoter anymore. But this is immature because the introns are still there. So after the splicing remove those introns, and the mRNA is formed, and after the mRNA is formed, it give origin to the amino acid chain and later to the protein. So this splicing part is really important because this is the process that remove all the introns that are in the DNA. And there are different splicings in the gene. So this is the most common way to have a mRNA, that is the immature mRNA. Basically, those black lines here represent the introns. But as you can see, after the splicing, here each color represents an exon. So after the splicing, we end up with the 4 exons in the messenger RNA. But there are variation of that splicing. So here, for example, there is an skip between the exon 2 and the exon 4, and we have a different mRNA. And here, for example, the mRNA doesn't have this part that is in black of the exon 2, that is the red one. So you can see there are a lot of alternative splicing that with the same sequence of the 4 exons can give origin to different mRNAs. So that it's cause some variation in the mRNA, and that is gonna have an impact in the protein that it's gonna produce with this process. And that is so important because if we talk about Phaseolus vulgaris, Phaseolus vulgaris in total has been identified 27,000 loci, but there are almost 37,000 protein coding transcripts. And that difference of numbers is because we have, or Phaseolus vulgaris has 9,562 total alternative splice transcripts. So as you can see here, in total there are 27,000 loci that produce proteins, but there are some alternative splicing between them, and that is why in total there are almost 37,000 protein coding transcripts. So that it's why it is important to understand the alternative splicing. And if we talk about genes, usually we talk about alleles, and an allele is when we have 2 or more versions of a gene. So for example, here we have an ACR allele that produces the red color in the flower, and here we have the CW allele that produces the white color in the flower. And when the both alleles are the same, like in this case CR, or in this case that is CW, we can say that both genotypes are homozygous. But when they are like in a mix, here that we have the 2 alleles, these kind of genotypes are heterozygous. So that it's an important difference between homozygous. Remember that both alleles are the same but in one genotype have both alleles that are the same, and heterozygous they are different. And although we talk about alleles when we're talking about coding regions or genes, in not coding DNA, we can use the same term of alleles if there are some difference in the sequence. So now that we mentioned a little bit about the genome, or we talk a little bit about the genome, chromosome, and genes, I want to talk about mitosis and meiosis and crossing over because they are really important in the breeding programs. So the mitosis occurs just in somatic cells, and basically start with one mother cell and ends with two daughter cells that are identical to the mother cell. So it's just a cell division, and this process is so important for development and cell replacement and regenerations and other processes. But as you can see here, there is not a genetic variation. So uh, the mitosis starts with one mother cells and ends with two daughter cells that are exactly the same. In the other hand, we have the meiosis, and the meiosis starts with one uh, cell that has a chromosome, a maternal chromosome, and a paternal chromosome. And here, before the first division, because meiosis has two divisions, so before the first division, there is the recombination, and that recombination it's so important because that causes a genetic variation in the progeny. So that is why in breeding it's so important to understand this concept. So before the first division, they have this recombination, and we produce two cells that are diploid as the mother cell, but they are not the same. So they got this recombination here, and as you can see here, one chromosome it's different to the. It's not the same, neither the maternal or the paternal chromosomes. And then they have a second division, and that second division produce in total four cells that they are haploids, and they are haploids because they are sperm or eggs, so they produce the gametes for sexual reproduction. And you can see here that two of those cells are different or like a unique combination, and that is why the meiosis it's important in sexual reproduction because it produce a genetic variation uh, when they produce the gametes. And the recombination it's really important in all the studies that we use in plant breeding and when we use molecular markers. For example, here just imagine that we have two molecular markers. We have the molecular marker A and B, and there is a recombination here. So if those markers are really close, we just cannot uh, watch or we cannot see that recombination here. But when we have two markers and the recombination occur between those markers, we can see or assess or follow that recombination with the molecular markers that we have in the genome. And easy way to try to understand that concept is when we create a population. So just imagine that we have those two parents and we cross those two parents, and then we produce a F8 population. So that F8 population uh, it's gonna have a lot of individuals with different recombination between them. And if we have different molecular markers across the genome of those lines, we are going to be able to identify maybe molecular markers that have association with the phenotype that we are interested on. And the last part that I want to mention or talk about is DNA sequencing and GBS, genotyping by sequencing. So before to mention or start to talk about DNA sequencing, I just want to mention that there are a lot of molecular markers that we can use in plant breeding, but like all these markers actually are really laborious in the lab, and that is why nowadays it's pretty common to use SNPs because it's pretty fast to identify the SNPs. The SNPs are really abundant in genome. As you can see, the SNPs are different. SNPs stand for single nucleotide polymorphism, and they are just different in one base. So as you can see here, there is just a change between guanine and cytosines, and here in this is the sample 1 and the sample 2. I have a change in the same nucleotide, and now I have adenine and thymine. So that it's an SNP, it's a like single differentiation in one nucleotide. And there are, as the variation is that small, uh, so they are pretty abundant in the genome. And to discover the SNPs in the genome is pretty easy. Basically, we just need to extract the DNA. In common being, we usually use the left of the of the plant, and we extract that DNA, and then we have to prepare the library. And basically, to prepare the library, we have to cut the DNA in small pieces, and we have to send those pieces uh, into a sequencing machine, and then we're just going to receive the sequencing data, or the sequencing data into the computer, and we have to start to work with that data. So uh, as you can see here, although this process seems a little complicated, when you have a really good pipeline in your lab, it's a kind of a straightforward process to do. And the good thing with sequencing is that the price started to drop in 2007, and nowadays it's sometimes it's cheaper get the sequencing data than the phenotypic data. So that is why, and that is really important for a genomic selection. If you want to use genomic selection in your breeding program, uh, always the price is a good thing to take into consideration. So if generate the genotype is cheaper than the phenotype, so genomic selection maybe it's a really good alternative. And there are a lot of different technologies to get sequencing data. So here we have the amount of information that all these technologies produce, and here the read length. Basically, the read length is the sequencing length that produce each machine. And here we have a Illumina technology. Illumina is pretty popular because the quality of the reads are pretty good, and the amount of information that Illumina produce, it's kind of high. If you have a if your target is just identify the SNPs in your populations, Illumina is good enough. If you are interested in do other kind of research, like for example, do de novo samples, so maybe PacBio or Nanopore are more interested for you because they produce a longer read lengths. So that it means that it's gonna be easier get an de novo sample with those with these methodologies. And now that you are a little more familiar with sequencing data, I just gonna give you an idea about GBS, because I know that some of you are working with GBS, and basically in GBS we have four big steps. So first we have to cut the DNA in small pieces. As I told you, that is the way that we produce the library, so we have to reduce like the complexity of the genome, and for that we use a restriction enzyme, and with that we cut the genome. And then the second step is that we have to put together those reads or DNA sequence with barcodes, and those barcodes are the IDs or the identifiers for every single sample. So we are going to put a unique identifier or ID for all the reads of a that come from the same sample, and some adapters to use in PCR. And then we after we put together the adapters and the barcodes to every single read, we can put together those samples in one tube, and then we can send that a pool of genotypes into the Illumina sequencing or the sequencing machine, and we are gonna generate all the reads with the barcode. So here the most important part are the barcodes, because basically the barcodes are kind of the name of every single sample, and when we have this big file we can split that information in smaller folders that are gonna contain the information for every single sample, and then we can use that information to identify the SNPs in the population. So as summary, I hopefully you have in your mind what is the big picture of genomic selection. I think that is really important that you remember that genomic selection is just another tool that we can use in plant breeding. It's not gonna remove conventional breeding, it's just another tool. Um, that the genome in common bean is kind of small, and in total we have around 27,000 loci that produce different proteins. So remember that as we have a common bean has alternative splicing, in total there are almost 37,000 protein coding transcripts. And the meiosis is so important, because meiosis is the way that in the breeding programs we produce a genetic variation between the populations. And if you are interested in genomic selection, the price of the DNA sequencing is so important. And one way to reduce the price of DNA sequencing is use GVS, basically because with GVS we are able to combine several samples in the same sequencing machine, in the same lane of the sequencing machine, and that it's going to reduce the price of the sequencing. And at the end, I just want to give you this website if you are interested in other genomes, or if you are interested particularly in Phaseolus vulgaris or other Phaseolus species. You can go here in Phytosome and you can find information about the genome, the chromosomes, the genes, and a lot of more information that I didn't discuss in this presentation. So this is the end of this video.